Welcome, everybody. My name is Julian Saba. I'm with the proteomics team at Thermo Fisher Scientific, and I will be doing the introduction to our first speaker, Max Crispin. Max reads biochemistry at the University of Oxford as an undergraduate. After doctoral studies at Oxford and Scripps Research, he went on to run the Glycoprotein Therapeutics Laboratory within Oxford's Department of Biochemistry. Max moved to University of Southampton in 2017, where he is the Professor of Glycobiology. His lab operates in the nexus of glycoprotein engineering and Glycan analytics, and is currently dedicated to the design and analysis of glycoprotein-based therapies and vaccines. Max's laboratory is funded by the Bill and Melinda Gates Foundation through collaboration for AIDS vaccine discovery, the International AIDS Vaccine Initiative, the U.S. National Institute of Health, the European Union Horizon 2020 program, and by Against Breast Cancer. He's currently supernumerary fellow at Oriel College, Oxford, and professor adjunct in the Department of Immunology and Microbiology at Scripps Research. Thank you, Max.
So thank you very much for that kind introduction. It's a real honor to be here today. I've been invited to talk about some of our work in virology, and in particular on the glycosylation of envelope viruses. And I'm really going to draw on a lot of our work from HIV, and of course, link that to all the developments in the SARS-CoV-2. So, one of the reasons we're so interested in glycosylation is that it's an extensive modification of envelope viruses. So these are the envelope spikes of a range of different viruses. Now, the grey platform, if you like, represents the lipid membrane of these viruses, and you have these attachment spikes sticking out of the virus. Now, these are critical for the biology of the virus. This is what mediates the attachment to the target cell and ultimately fusion of the viral membrane to the target. Now, in blue, you'll see all these carbohydrates. Now, there are different types of carbohydrates. Today, I'm going to be principally focused on N-linked, so asparagine-linked carbohydrates, and they're attached by the host cellular machinery at a particular sequence within the primary structure of the protein. Now, this is important because that means the location and attachment points of these glycans are actually genetically determined by the virus. And what you'll see is that we're very interested in this in terms of their wide range in biology, but in particular, the fact that they can be used as glycan shields. Now, I really like the image of the sort of wolf in sheep's clothing, because this really represents the idea that the viral spikes are covered by sugars that are ultimately self. So your immune system really doesn't see them as a foreign viral entity at all, but rather self structures. This means they can obscure large parts of the antigenic surface of the virus. Now, there are a couple of exceptions to this. So if you have viruses that are zoonotic, so they're moving between different organisms. So, for example, a flavivirus, maybe insect-borne, they'll have sugars that are insect-like. And you can have antibodies against these types of sugars, and it can act as a bit of a barrier to transmission. And this is even the case in humans, where we have blood groups. Now, the base of blood groups are carbohydrates, and you have these particular modifications that are specific to people that are going to occur on the glycoprotein, but also the glycolipids. And this can, even in HIV, give a measurable hurdle to infection. Not very significant, but measurable. But for our purposes today, we can largely think of these sugars as self and offering a limit to how antibodies can ultimately neutralize viruses. Now, in the context of HIV, it's been really sort of interesting how the field has been slowly, increasingly focused on the carbohydrate shield. Now, in HIV, you have a virus that's infected a patient, and it's sort of hiding and resident within that patient for many years. So that has particular implications. It means that the virus has to survive under the selective pressure of the host immune system, and that drives a very high density of these glycans here shown in green. Now, the reason why the vaccine field is so interested in these sugars is that over time there have been about a third of patients develop so-called broadly neutralizing antibodies that can often recognize the carbohydrates, or at least have evolved to avoid them as well. Now, we say broadly neutralizing because HIV, the mutation rate is phenomenal. And so, these antibodies are actually capable of neutralizing a range of different viruses, regardless of the mutation. So they're incredibly broad. And here we show a remarkable structure by Ian Wilson's laboratory at Scripps. And this shows one such antibody, the PGT128, recognizing the target HIV glycoprotein. Now, the remarkable thing here you'll see in the antibody in blue is that it's recognizing the carbohydrates, in this case, two different glycans, we call them, attached to two different asparaginins, and it's slotting in between these and making only minor contacts with the protein surface. And what you'll see is actually those two little arrows, the blue and the green, indicate a beta sheet has formed. So even the contact of this antibody to the protein is not really driven through side chains, but through the backbone. So this means that mutations that are occurring in the protein are really not impacting its binding, and therefore it can neutralize all these wide range of strains. And this is even true for glycans. If you were to mutate some of these glycans, often the antibodies can just change their angle of attack and bind to a neighboring glycan. So it's really, these things are have remarkable properties. And they've now, as a field, identified numerous broad neutralizing antibodies that cover almost the whole surface of the HIV spike. And here showing this nice little rainbow plot of a variety of different classes of these antibodies and their carbohydrate targets. Now, why isn't this a solution for a vaccine? Well, the problem is these antibodies take a long time to develop. So whilst they can, if passively transferred, offer protection, if you're already infected, they cannot eliminate the infection because the virus is too diverse already. And, their mutational history, because they are evolving to recognize self-structures, means that it takes a very long time for antibodies to evolve. So the real challenge then is to be able to design ways that you can elicit these antibodies to target this. Now, our involvement is very much on helping the vaccine community in studying the carbohydrate modifications of their immunogens, because if you're trying to make your immune system recognize these carbohydrates, your immunogens, your vaccine candidates, they ought to present those carbohydrate epitopes in order to potentially elicit these types of antibodies. So monitoring immunogens is really, really important. Just stepping back a bit about more of the biology, the glycosylation impacts a wide range of different features of the virus, but also we can learn a lot from recombinant mimics and studying these recombinant immunogens. So, for example, the glycan processing of immunogen can impact can inform about the integrity of the protein. I'll illustrate a few examples of that. It can also impact the trafficking of the immunogen through the sort of lymphatic system and ultimately interaction with your immune system. So they can impact the sort of immunogenicity of the protein. And we've mentioned about the presentation of actual carbohydrate epitopes. And we can also learn a little bit about the degree of glycan shielding that's occurring across these virus classes by studying the processing. Now, referring to the biology, glycosylation impacts extensive areas of viral biology. It permeates a lot of the interactions between the virus and the host. Now, here we've just included one example of a viral glycoprotein being manufactured by the producer cell and incorporating the virus. And you'll see a recurring theme here is I'm using a color code to show early sugars they're placed on the ER, showing in green. These are sort of oligomannose forms. And as the protein gets produced and secreted through the Golgi, you get more and more elaborations, and we're depicting those in a magenta color to illustrate, processing. And you'll immediately see in the example we've chosen that viruses have a mix of this green and magenta, meaning some of the sugar structures that are present in the ER are getting produced and actually added onto the virus. Now, this is a really quite important feature because your immune system is geared up to recognize these mannose structures. Yeast have mannans, chemically the same thing. You have host lectins designed to recognize mannose as a sort of inflammatory signal. And it's really interesting that in mice that have been engineered to display mannose on their cells, they have chronic activation in the innate immune system. So there's a big pressure on your own glycoproteins not to display mannose, whereas in viruses, they have such a high density of sugars that this leads to these mannose features appearing. And in fact, it's a double-edged thing. In one way, the appearance of mannose can be used to recognize and potentially neutralize viruses by the innate immune system, but they can also be exploited by the virus in infecting those very cells that display those lectins. So it's a really quite complicated area of biology. So just to reiterate in terms of the pathway, because we will be showing the sort of some of the sugar structures present on these glycoproteins. The proteins are made within the ER. Now, one of the selective pressures for viruses to have lots of sugars is to help folding. So there's a folding pathway that utilizes the sugars within the ER. And then you get this series of reactions, trimming of mannoses and building up of more of other residues like galactoses and sialic acid. And we describe those as sort of complex type glycans. Now, importantly, and this is an example from my early antibody work, you can trap the biosynthesis artificially using inhibitors or mutants. And we've studied here the sugars by just basic mass spectrometry here, and it shows the homogeneity of the early stages of this pathway. And it's only really when you get to the Golgi that you get this complexification occurring. Now, returning back onto what we can learn about studying glycosylation of recombinant proteins, and this is where I begin to include some of the work on the SARS-CoV-2, we've already, and this is remarkable to me, there've been identified antibodies that have been identified that can actually recognize carbohydrate components. So here on the left, you'll see an antibody that recognizes both the protein, but also a fucose residue present on the viral glycoprotein. Now, this is quite interesting because what this means is that recombinant proteins are being able to have carbohydrate processing events that can be recognized by the host immune response. So we already have a pretty good indication that recombinant systems can capture some of the features of authentic infectious viruses. And in fact, there's already been quite a few studies, I've highlighted a couple here, showing that in the SARS, there's been the viruses present a mixture of complex glycans and sort of oligomannose ones. Now, this is really quite important because it wasn't obvious that this was going to be the case. Coronaviruses actually bud into the secretory system rather than budding from the plasma membrane, like is the case in HIV. So what impact could that have on glycan processing? And actually, the evidence is very strongly that you do get pretty normal maturation events. And I've made a little note here that I will be showing some remarkable data by Tsai Lee and his work on the native authentic virus. Very excited to see that. So the other thing, of course, is I mentioned that the recombinant proteins, when used as vaccine candidates, their glycosylation state can affect the trafficking of your glycoprotein within your immune system, and that is obviously intimately linked to the elicitation of an antibody response. And that's been beautifully demonstrated by Darrell Irvin's group in MIT, and that was published in Science in 2019. So what lessons can we learn from HIV? Now, what we have there is a lipid virion with only a small number of spikes protruding, but these are sort of trimeric spikes, heavily glycosylated. And what we've shown, and here is just some UPLC data of fluorescently linked of glycans that have been released from these glycoproteins. What we and others have shown is that native-like glycoproteins actually present more oligomonos. So at the top is just the GPON20 subunit, and that has like a third oligomonos. If you artificially trimerize these glycoproteins in this sort of pseudo-trimer, you basically don't lead to a big elevation. It's only when you have native-like folds where you get this elevation of oligomonos. So that's sort of what I was talking about this relationship between protein integrity and protein conformation and the impact on trimming. Now, we've been investigating this on a sort of site-specific level, doing glycopeptide mass spectrometry, and our first when we first dipped our toe in the water in this was in some work published in 2016, so comparably recently. And at the time, the HIV literature was a bit confusing. So depending on the methodologies that were employed, you'd get a different sort of readout of quantitation. And, often that wouldn't quite agree with what we'd know about the epitopes based on neutralization assays with carbohydrate-specific antibodies. So I was a bit nervous about how to approach this. So we went back to some pretty old-fashioned methods. We isolated glycopeptide fractions by reverse phase HPLC, and we studied those fractions of the HIV glycoprotein by different methods, MALDI, chopping the sugars off and analyzing by UPLC, and of course, inline LCMS methods. And we settled on using an Orbitrap with protein metric software, and that gave really nice agreement to a lot of these sort of slightly more traditional, established methodologies. And here we show just an alignment between just MALDI of these isolated glycopeptides through and the LCMS. And what this enabled us to do is have confidence to roll out the full LCMS, ignoring these more labor-intensive methods. And this is the methodology we've been using. What this has enabled is plotting the carbohydrate processing states across the virus spike. And this is, again, HIV. And you'll see in green these mannose patches that occur across the surface. And also hotspots of highly processed areas, shown in magenta, these complex glycans, and areas where there's an intermediate level, shown here in orange, where there's a degree of under-processed, a mixture of mannose and complex. Now, we can also extend this to, looking at those different formats, the monomers, the pseudo-trimers, and native-like trimers, and understanding how, on a site-specific level, does the adoption of these native-like folds impact the processing states. And plotted here is a sort of percentage point difference. So we're just doing, just displaying the changes in the percentage of oligomonos between monomer and trimer on the left and pseudotrimer and trimer on the right. And what this indicates is where there's red, in the trimer, there's been a big increase in the amount of oligomonos, so more a limited amount of glycan processing, and that's pretty similar in the trimer, the pseudo-trimer as well, just to a slightly lesser extent. Now, this can be rationalized structurally because we have, thanks to Kelly Moorman and others, crystal structures of monosadases, and this is from the Moorman lab, in complex with their ligands. So in cyan, we have the outline of the crystal structure of the monosadase 1 in complex with its target. So this is a deactivated enzyme in complex with its target. And what we can do is superimpose that complex onto the HIV surface, and we see the extraordinary degree of clashes with neighboring sugars. And in fact, it envelopes all three neighboring sugars in this particular case. And that also it got important biological consequences as well, because we know if we do knockouts or the virus naturally loses a sugar, it doesn't always lead to a big impact on processing. They often stay oligomannose within the oligomannose patch. So that is also important when we consider about epitopes for broadly neutralizing antibodies, is that there is a certain redundancy built in to the system, and even if the sugars are appearing and disappearing, it can have quite a minimal impact on the processing state of these surrounding sugars. But there does seem to be a bit of a contradiction here. I'm saying that mannose is appearing because it's not accessible to enzymes, and yet it can have this wide-ranging impact on interaction with lectins and even with antibody recognition. And here, shown on the left, I'm trying to put this on the same basis, we have the mannosides, you'll see entirely enveloping its target glycan. This is quite important because enzymes active against carbohydrates are often pretty linkage specific, and they need to have quite extensive recognition interfaces in order to achieve that selectivity. And this is in complete contrast to a lot of the lectins shown in the middle, where they can recognize the tips of sugars and don't need to embrace them quite so much. And similarly with antibodies, they're capable of slotting, as we've seen before, slotting in between them and a variety of different mechanisms. So really, the virus can be in a situation where it's added so much sugar that it can't get processed by the enzymes, but it's totally accessible to lectin recognition and to antibodies. Now, what are the implications for SARS-CoV-2? Now, that similarly has these viral glycoproteins protruding, and as soon as the outbreak became apparent that this was going to be a pandemic, we began to go on a sort of coronavirus response level. And I would really like to acknowledge the support I've had, and we've all had, from our corporate partners, including Thermo, and of course the flexibility of our funders, so which really enabled sort of slightly out of program work. And this extended from explicit use of financial resources, also the redeployment of staff. So that's been incredible. So we've been not only analyzing immunogens, vaccine candidates, but we've also been generating glycoproteins to set up serological testing, particularly with the University of Birmingham and the company binding sites. So big thank you for everybody there. So we first contacted Jason McClellan's lab at the University of Texas and got hold of, where he sent us a batch of protein of the viral spike, and also with the expression plasmid so we can make it locally. And our team, I like this slide because it shows not only some happy students here, so Yasu in the middle, who did a lot of the work, and also Joel Allen with his thumb up. I'll highlight a lot of the work in the forthcoming slides, but also it indicated our transition from the University of Oxford to University of Southampton, and the HRE work I present is actually performed in the Glycobiology Institute. So what I'm going to be talking now is our analysis of this SARS immunogen. And I'm just going to give you a little bit of just technical information here, just to understand some of the language. So these I'm going to be talking about oligomannose glycans, I've already mentioned. And here I'm showing them as sort of green mannose residues, and you'll see that they're attached to two GLUTNACs. We call those oligomannose glycans. But you'll also see in magenta the complex glycans, and this gets a little bit more involved. But with the mannose ones, we call them MAN9, referring to 9 mannose residues, to MAN5. And in contrast, the description of complex glycans gets a lot more involved. And in this talk, I'm going to be largely simplifying it, but basically you can have multiple branches and multiple antennae. And so in the figures, you'll see we talk about A2 or A3. This is the number of antennae and branches, basically, that the glycan has, and it's also capable of having galactose or sialic acid. We often simplify this by classing them based on the number of branch points or the fucosylation states. And I can just give you one example of that. Here, you get these biantennaries, but with potentially with or without a bisect or with or without fucose. And similarly, you will see the odd situation where we have hybrids, where the mannose trimming hasn't occurred fully, and yet it can also pick up some of the features of more complex glycans, and hence why it's referred to as hybrid. Now, the analytical methodology we use is obviously using the Orbitrap for the site-specific analysis of the glycopeptides. We tend to do a lot of offline UPLC analysis of fluorescently labeled glycans, and we also do quite a bit of characterization of released glycans using a SYNAPT. And it's these methodologies we combine as part of our sort of analytical workflow. We're focusing mainly here on the site-specific analysis. We adopt a very traditional proteomics approach of trypsin and chemotrypsin treatments. And again, the data I'm showing is generated on the Orbitrap Fusion using protein metrics software. So many of you will be aware we can generate a chromatogram of our glycopeptide signal. We search for peptides modified with carbohydrates. We identify them, check for the isotope distribution. We're checking for the fragmentation signals, again, across different charge states. We're then integrating the chromatogram, and it's summing all those different forms, for example, charge states. We sum all those together, and at that stage, we can actually begin to get a really good quantitation shown in these bar graphs at the bottom. And this particular site taken from HIV, you can see heavily dominated by Man9. Now, switching to SARS-CoV-2, I'm going to show more complicated data, and this is what we tend to see. And you see all the different glycan sites, and other, but I don't expect you to see, it's just load numbers. These are different types of carbohydrates. You can see very complicated. One of the take-homes here is that glycosylation is highly heterogeneous. It is complicated, and it adds a big burden on investigators to actually try and present the work in a simple way. And I hope we go some way to achieve that. But the other thing I want to point out is you'll see, if the eagle-eyed amongst you will see error bars here. And this is actually data from 3 different protein expressions, including one from Texas and one from Southampton, and it's remarkably stable. So we can consider glycosylation as highly heterogeneous and tempting to say random, but it's a reproducible randomness. So, that's a really important take-home. Now, taking that complexity, we can again simplify these, just looking at the number of branch points and the fucosylation status, and you can begin to see the distribution of sites which are presenting mainly oligomannose glycans, and an extensive range of ones which are presenting these magenta complex and highly processed structures. And you're probably already noticing that there's an elevation of processing compared to the HIV equivalent. And we're going to again map those processing states onto the cryo-EM structure. And you'll see that unlike in HIV, we don't really have a mannose patches, say, with these very dominant, exclusively oligomannose sites clustered together. We have snippets of that going on, but the take-home message is it's largely processed. But this representation using this categorization system does lose some subtleties here, because if you put it on more of a sliding scale of mannose content, you do see that it is presenting some mannose, and that's important because that degree of mannose can influence immunogen trafficking, etc. So, we are seeing a degree of mannose appearing. So there is still a bit of a deviation away from typical mammalian glycosylation. Interestingly, we do see the features targeted by antibodies against CoV-2, like the one identified that's specific for the fucose. We can also plot the abundance of other modifications, like fucose in the middle here and sialic acid. And you'll see that inverse correlation, as you'd expect, where the sites which are display less oligomannose are displaying enhanced levels of fucose and sialic acid. But taking the mannose level one step further, we can begin to compare across the different viral classes. Now, the 3 panels on the left, we have the original SARS-CoV-1, the data I just showed, the CoV-2, MERS, and then for a comparison, we also included Lassa fever and HIV. And you can see that the coronaviruses are actually landing at the lower end of the spectrum here. So when we first thinking about this, we really, thought the take-home message here was that this showed a degree of vulnerability, that these glycans were not as dense as in HIV, and this probably reflects a different biology, like it's sort of hit-and-run virus. It's infecting things, a bit like flu. It's ripping through a population rather than having to stay, which is HIV's MO, if you like. Now, we can begin to also relate this processing state that we can measure with mass spectrometry to cryo-electron microscopy. And this is some beautiful work performed by Zach in Andrew Ward's lab. I've shown his email here. And he's been able to plot signals using cryo-electron microscopy of the carbohydrate, even if it's diverse. And so you see on the left in HIV, you have extensive range of this like connected network of signal you're seeing in cryo-electron microscopy. Whereas you compare to the SARS, and this is the SARS-CoV-1, you get much more isolated, less sort of regions of connected carbohydrates, so much more pockets of individual ones. Again, reflecting that slightly more dispersed carbohydrates, less shielding, and ultimately quite an extensive range for antibody recognition, so big areas of vulnerability. Plotting that together, we can look at oligomannose abundance across the viral spike and looking at a measure of glycan density. And as you'd expect, there's a pretty close correlation between increased glycan density and the appearance of these oligomannose structures. Now, a lot of this I've shown has been in recombinant systems, and I tried to reassure you that they do capture many features of the native virus, but that has been greatly enhanced by just breathtaking work of Sai Li's lab in China. And they've done some beautiful work working on authentic infectious virions, doing the cryo-electron microscopy, showing the architectures of not only the spikes, but also the underlying nucleic acid. And as part of that work, they're able to analyze the carbohydrates of the coronavirus. And here, using a slightly different color scheme, but basically blue is the oligomannose structures, you can see that, again, the coronavirus is naturally processed. It's not, processing isn't impeded by this unusual budding pathway. And that where you get mannose presented in the recombinant system, that is captured to a degree in the native virus. Now, what's interesting to note is that it's all shunted down, so there's more processing. And interestingly, we see a similar pattern in HIV where, and this is again UPLC analysis of virally derived GP120 and then GP120 from recombinant systems. And you'll see that whilst the recombinant system captures the main features of the viral glycosylation, there is a tendency to slightly enhanced processing. So what we can do is also do that, plot that site specifically. And here, the bar graphs are just individual sites showing the amount of mannose versus not. And you can see the two structures on the left from the recombinant show extensive oligomannose. And in the virus on the right, where you have sites of intermediate processing, these are generally driven to completeness. So you still have oligomannose features, but probably the longer residency time has led to increased processing. But overall, what we can conclude from this is that monitoring the glycosylation of recombinant material is really important in understanding your immunogen, to what degree it matches the target virus, and of course, to what degree could those modifications influence the behavior of your immunogen when used in the clinic. And there is a prospect of engineering it artificially. So that's also a really interesting line of research. So this brings me to thank the people that have been heavily involved in the work during the coronavirus. We interact with so many labs across the world that are in the, I'm embarrassed to not be even able to include everybody, but big thank you to all our co-authors and collaborators. I've highlighted here some people who had particularly large influence on the work presented throughout this presentation. And you'll see in the picture, Joel Allen saying hi to Yasser in a suitably socially distanced way. So big thank you to everybody who listened today, and I very much look forward to your questions. Thank you.
Thanks for that excellent talk. I'm glad to see somebody who's done glyco work in the past. Glycan's getting mainstream at the moment. So yeah, it's really excellent to hear your talk here. So we do have time for questions, so feel free to type in the questions in the chat box. So the first question that I have here is, you mentioned about the importance of glycan processing, but how important is it that sites are highly occupied?
Oh, that's a very perceptive question. Yeah, so one way of answering that is that we don't know in many cases, but we're getting a very clear idea of the importance of that actually in HIV vaccinations. Now, what we see is because the density of the sugars is so high, and often it's those regions where we want to try and tease out an immune response towards, when there are sugars that are absent, it creates these sort of, the terms glycan holes, and these turn out to be highly immunogenic. Now, this is not a good situation, if you recall back to the last slide, where we noticed that in HIV, the occupancy of the viral glycans is incredibly high. So the recombinant systems do often pick up holes. It is largely, we think, driven because you're making it as a sort of soluble expression-enhanced version. And there's a lot of effort in actually re-engineering these HIV trimers so that they don't have these holes. So one line of work that we've been doing with Roger Sanders, Ronald Durking, Joel Allen, to name a few, has been to try and re-engineer the primary sequence of the viral spikes in order to enhance occupancy. Now, the occupancy rates can be influenced by the precise sequon. So in a simplistic way, NXT is slightly higher occupied than NXS. So one way is simply to mutate those S's to T's, and you do, if you use that approach, get a very high degree of occupancy. Interestingly, because the density of sugars in HIV in the sort of primary sequence is so tight, you can have a rapidly, or they're so spaced so close together. It does create some complications. So if you over-enhance the occupancy of one, you can have detrimental impacts on neighboring sites. So actually what, particularly Ronald has been working on is winding back some of this occupancy to maintain high efficiency throughout those regions. And what we see working with Andrew Ward's lab at Scripps using cryo-EM is that we can see those antibodies that recognize these holes being eliminated by this. So that's another example where mass spectrometry measurements of occupancy actually match other structural biology questions. But yeah, great question. Occupancy is emerging as important in particular applications, definitely. Thank you.
Great, thanks, Max. This next question here is, does the mannose content change in different cell lines, such as CHO, Chinese transport?
That's a really important thing. We get asked this a lot because obviously the CHO cells, the Chinese hamster ovary cells, are such an important platform for production of biologics. Whereas a lot of researchers can be using the 293F system, for example. So it's totally understandable to be concerned about how those systems are related. But one reassuring thing is it depends on what you're trying to optimize. Now, in HIV and in many of these viral glycoproteins, the mannose content is really quite a key quality indicator and quite dominant in terms of its biology. So when we look at that, the two systems are highly similar. And it probably reflects the evolutionary conservation of the enzymes themselves. But, actually, the mannosidases are conserved, all the way throughout the eukaryotes, basically. So they behave very, very similarly. Now, I would just add one other consideration here, and this is a bit subtle, but you can have mannose appearing in situations where you're expressing to an incredibly high level. And so many of the audience will be aware of the successes people have had in production of antibodies, where they now can exceed a gram a litre. And, in those types of systems, you can begin to get sort of product inhibition. So you're basically hydrolyzing loads of mannose off your, in this case, antibodies, and the mannose within the ER actually adds as an inhibitor. So mannose can begin to appear in certain situations when you get to incredibly high yields. But in these types of situations, they're all very comparable. So I should obviously not leave it just on mannose. When you look at the complex glycans, they do change quite to a large degree. So the degree of branching, the level of sialic acid, all of these types of parameters do move. So depending on your application, it can be important. So thank you.
Okay, thanks, Max. The next question is, to what extent does mass spectrometry data agree with 3D structures of viral glycoproteins being determined by cryo-EM?
Yeah, that's a really interesting one, because a lot of the structural biology work that's occurring is revealing the architecture of carbohydrates. Now, the last slide I showed was this mapping of the carbohydrate cloud, which is really trying to take signals from almost like the disorder and plotting the disorder, and that gives a sort of indicator of how these sugars might be interacting. But that's quite a special case. You used to study these, the wider heterogeneity. In many cases, structures have been isolated where the reconstructions revealing the architecture of individual glycans in quite amazing detail. Now, it varies considerably. In some areas where they're on extended loops, or they're flexible, you don't see that same resolution by traditional methods. But in cases where there's a high glycan density, or is bound in complex with the target antibody, the cryo-electron microscopists and even crystallographers, both of those methods are yielding very precise structures of glycans. Now, I actually showed in one of the slides a complex of an antibody with a target glycoprotein, and that was entirely by crystallography, and revealed all the branch points. So where we're seeing sort of structural homogeneity, we see really strong agreement. And where mass spectrometry comes into its own is when it's highly heterogeneous, and that's where we start to lose information in some of the structural biology approaches, and where mass spectrometry has such a huge role to play. So thank you.
Thanks, Max. Our next question is there any evidence for O-link glycosylation in the current coronavirus SARS-CoV-2?
Yeah, that's an interesting one. Obviously, what I've been talking about today is heavily dominated by N-link glycans. Now, there's a couple of reasons for our emphasis. One is that there's an awful lot of them, and also that you can very easily see in the primary sequence their attachment. An attachment point is encoded. O-links is a very different proposition. There's no defined attachment sequence, so you can sort of broadly categorize into two areas. We have regions which might be O-link modified, which are heavily dominated by serines, threonines, and proteins. We don't see that type of sort of mucin-like domain within SARS-CoV-2. So what you got left with are modifications that occur on slightly more globular regions or potentially extended loops. Now, we did have a look in a few places where our data enabled us to, and we saw evidence of very low levels of O-link glycans. And other investigators have found, a similar story. So very low levels of modifications. In places, people do see more, and some of those are methodological differences, and sometimes there's sample differences as well. So an important theme is really, it's very tempting to compare the analytical endpoints and to sort of weigh up who says there's more or less. But we also need to do the same level of scrutiny on the samples, and basically different samples with different, how do I put it, oligomeric states even, will tend to pick up different levels of modification. In HIV, we've looked at one particular area where O-links have been reported and actually see a big elevation of that modification in structures that are sort of pseudo-trimeric. They're not actually adopting the native form. And when we express these stabilized native forms, the degree of O-link modification drops. So, yeah, the jury's slightly still out on this. I think they are there. Their role and their abundance is still being refined, I would say. So thank you.
Thanks, Max. Is there a role, is there a biological role for all the glycan heterogeneity you see?
So it's quite tempting to sort of, understand glycan biology by, if you have a particular sugar, you're going to get a particular effect. And, hence why we're so obsessed with the amount of mannose or potentially amount of particular sialic acid linkages, which can have immunological impact. The role of heterogeneity is really interesting. I would say the big, when we look at the wider biology, one thing is there's a massive redundancy within glycosylation. So just because you see a particular linkage and particular structures in one situation doesn't always mean it has a particular function. And famously, you can do mouse knockouts and knock out particular linkages, and you only see aberrant pathologies picking up in isolated areas in many of these sort of terminal elaborations. So, redundancy is a heavy, as a particular hallmark of glycosylation. Now, in case of HIV and with other viruses, there may be a role. I mean, one of the advantages of heterogeneity, if you're being targeted by antibody recognition, is that it sort of dilutes down the amount of epitopes you're presenting. And in the case of HIV, we see examples in neutralization assays where an antibody that binds sugars is incapable of fully neutralizing a particular viral isolate. So that means there's no genetic variation there, but there's variation actually in the virus. And sure enough, that can be assigned to variations in carbohydrate, to the heterogeneity within glycans. So it's a sliding scale of importance. You'll find examples where the heterogeneity literally reads out in terms of its impact on neutralization. And you'll have other examples at the other end where heterogeneity is just a byproduct of the evolutionarily selected pressures on glycosylation generally, and potentially in very different parts of the body and different cellular systems. So hopefully that's not too elaborate, complicated answer to that question.
No, that's perfect. Our next question is, how can therapeutic target a native spike protein if the recombinant targets display exaggerated glycosylation?
That's an interesting one. So what we'd first have to establish whether glycosylation, the details of glycosylation matter at all. Which is probably a controversial thing to say, having just lobbied about their importance. But some vaccines may be particularly potent without sugars at all. So E. coli-derived RBD domains may be effective in certain settings. So where glycosylation comes into play is when you need to optimize something that's not quite working, or you want to have an enhanced form of the vaccine. So certainly second generation approaches, this is where you need to put your full attention. And there, the details will it's slightly empirical. Does the lack of, for example, if you're making a protein that has no mannose on, does that limit the trafficking routes to actually generate a type of immune response that you'd like, and the potency of immune response you'd like? So the degree of matching in many systems is unknown. In other ones, like in HIV, you want to match the epitopes actually displayed in your that are targeted by your broadly neutralizing antibodies. And there, I would say that the degree of matching and where is there agreement and where is there divergence becomes important. And thankfully, many of the antibody epitopes targeted by broadly neutralizing antibodies in HIV target regions where there is agreement rather than where there's divergence. So yes, again, it's a bit specific to whichever virus you're looking at.
Okay, thank you. Can certain glycosylation profiles be an artifact of specific expression conditions and not endogenous biology?
Well, almost certainly. So one of the reasons why I emphasize the mannose content is that's one bit which tends to be in close agreement. Not always identical, but in close agreement. As soon as you talk about other elaborations, then you're very much looking at your sort of model system, and what you can learn from recombinant material can be quite limited. So this, again, it just emphasizes where we try to take information and we think it is informative. Mannose levels, I think, are one of those areas, and where we don't pay so much attention to, where we know that's, gives you deep insight into the behavior of human embryonic kidneys, and it has less relevance to those other systems. So yeah, I think you need to use model systems eyes wide open. They can tell you a lot, but they also have their limitations, and that's really clear.
Okay, thank you, Max. You answered part of this question, so I'll ask the second part, which is the viral proteins, do they display O-glycosylation? Is it of importance if you see O-glycosylation? Or how they are different from the typical N-glycosylation? Is there specific roles that they play differently than something like N-glycosylation?
So let's just keep it more. Let's look at that within a virus context. I mean, if you're picking up modifications, say for example around protease cleavage sites, a lot of the proteolytic maturation events occur very late on in the secretory system. So if you're picking up O-link modifications that are somehow impeding that proteolytic event, that's going to change your, the abundance of your mature proteolytically matured product. So you can envisage scenarios where O-links do suddenly become quite important. If you see them in large abundance and other people don't, that may be a strong indicator that the protein conformations you're seeing are that much more diverse and flexible than many other systems. So I think it can be a really important indicator of the underlying protein integrity and conformational space, but it's clearly not always negative. They do happen in viral glycoproteins, and they're meant to be there. And so, you have to be very careful about the precise system that you're talking about, and they contain information basically. So yeah, keep an eye on them.
Great, thanks, Max. Again, thank you for this excellent talk and taking time to answer a lot of these questions that came in. Just to the audience that are listening in, we're going to take a 30-minute break, and we're going to come back about 30 minutes past the hour, where we'll have 4 more excellent talks focusing around virus research. We will also have a roundtable after the 4 talks that have occurred, where the participants from this presentations will join our moderator, Andreas Huber, to have an extended discussion around virus research. So again, thank you. Take the next 30-minute break, grab a coffee, grab a tea, then come back and join us for 4 more excellent talks. Thank you all, and thanks, Max.
Thank you.